everyone, here is our third video about protein synthesis. This one is all about transcription and translation. So how do we write transcription and translation in science class? Well, just remember transcription happens first. That actually happens in the nucleus where DNA is the template for mRNA. That mRNA then leaves the nucleus and goes out to a ribosome that's in the cytoplasm and does translation. Translation occurs when the mRNA codes for certain tRNAs to bring in specific amino acids that are going to get together and form proteins there at the ribosome. Those proteins can then fold up and be used in our bodies. But transcription, how do we write it? Well, first look at the DNA, then add mRNA nucleotides to the DNA template. So whenever you see an A in DNA, put an U in mRNA, because remember there's no T in mRNA. It just has uracil instead of thymine. So when you see an A in DNA, put a U of mRNA. When you see a T in DNA, put an A of mRNA. When you see a G in DNA, put a C of mRNA. And when you see a C in DNA, put a G of mRNA. Once you've made your mRNA strand by looking at the DNA and putting mRNA nucleotides with it, you're then going to do translation. So first what you're going to do is in your long list of mRNA nucleotides, you're going to look for AUG. When you find AUG, it is the start codon for methionine. Methionine is an amino acid, and that's always the start of a new protein. After AUG, what you're going to do is put a circle around the nucleotides in groups of 3. A group of 3 mRNA nucleotides is called a codon, and every codon will code for an amino acid. You can use a codon chart like the ones you see here to determine which amino acid is going to be called for, and when you find stop, that means you're done making your protein. There are certain codons that are just stop codons, and that means that's the end of the protein. So how do we use codon charts? Well, I'm going to show you how to use both of these types because both of them could appear on the SOL. This is a circle chart and this one is a square chart. They're named after their shapes. But let's say you have A, U, G. You're going to start in the middle if you're using the circle chart. Always on a circle chart, start right in the middle, then go outwards. So A, U, and G for methionine. So you started in the middle, you went A, U, G. You just follow those letters outward as you go and read your codon, and that's going to give you the correct amino acid. If you're using the circle chart and you said G, C, A is next, you would start in the middle, you go to G, C, A. A is for alanine. GCA is for alanine. You can see that there, and that could be abbreviated ALA. And next up, if you are doing UAG, you start again in the middle, U, go outward, A, go outward, G, and you see stop. That would tell you that's the end of that protein. Now, are any proteins really going to just be 3 amino acids long? No, but for the sake of practice, it is. All right, since you've seen the circle chart, let's look at this square chart. So AUG. Now we're going to look at the first base on this column, the second base on these rows, and then the third base is over here on my last column. So if I'm looking for AUG, I'm going to start with my first base, A. I see that A is right here, and I know then that my answer is going to be somewhere within this row, this big row. Next, I look for my second base, U. U is right here, and that tells me that my answer is going to be somewhere within this first column of the chart. So A is going to be in this row, U is going to be in this column, and then for my third base, I'm going to look over here, and I see G, and I follow it back, and I see MET. MET stands for methionine. Next, I have GCA. G is right here, and so I know my answer is going to be in this bottom row. I then would look for C up here. C is in the second column there, and so I would know my answers are going to be in the bottom row and in the second column. And look, actually all of the answers in the bottom row, second column are ALA or alanine, but just to be safe, I would see that I'm doing GCA. I see my A there, and I follow it and find alanine. All right, my last codon is going to be UAG. So I see U, I start in the first column. U is in the first row. A is in the third column. So I know my answer is going to be somewhere in this area, but also in this area. So this is the overlapping area. And then I find G over here as my third base. I follow it backwards and I see stop. So that tells me it's going to be a stop. So AUG is methionine or met. GCA is alanine or ALA. And UAG is stop. Here's an example of how to do the whole process. So you will see DNA, and that DNA was in the nucleus. It was in a chromosome that unwound to let this process start. And when it unwound, we had transcription. Now, transcription is when you use DNA as a template for RNA. And I need you to remember something. In RNA, we do not have thymine. Instead, we have uracil, which is a U. So be on the lookout for that. In our transcription, T would call for an A from mRNA. You would think A would call for T, but remember, mRNA doesn't have thymine. It has uracil instead. And so you get a U. C calls for a G. A calls for U, because those are all A's. G is going to call for a C. T is going to call for an A. C is going to call for a G. A will call for a U. C will call for a G, and T calls for an A. Then I scan my RNA and I look, is there an AUG somewhere? Because I know AUG starts at, there it is, AUG. And I would circle it. Then I would circle every group of 3 after there, because those are my codons. Those codons are going to then be used at the ribosome. Those mRNA codons code for specific tRNAs to bring specific amino acids to the ribosome that will then be put into the protein form and fold up to make proteins that work for your body. And what we can do here is I'm going to actually switch back and forth between the circle chart and the square chart on these to tell you which amino acids we have. So I'm doing the circle chart first, and I have A, U, G. Here's A because I started in the middle, A, U going outward, and G going outward. A, U, G calls for methionine or met. All right, I'm going to switch over to the square chart just to show you both. U. So my first base is U. I know I'm in my first row. My second base is U. I know I'm in my first column. So first row, first column. And then to find out which one it is it PHE or is it LEU? Well, I go over here and I see another U. It's PHE, which is phenylalanine. All right, I'm going to go back to the circle chart for the next one. I start in the middle and I see CAG. So CAG. And I see glutamine, which is actually abbreviated GLN. You might have thought GLU, but GLU is actually glutamic acid up here. So glutamine is something else as GLN. But if you're using the circle chart, you can write out the whole word and you'll get full credit. All right, and then last but not least, let's look at the square chart to do our last codon. So I have UGA, and remember U is going to be in the first row, and then G is going to be in the last column. So I know I'm looking in the first row, the last column. Which one is it going to be out of these 4? Well, I go to my A, and then I go backwards and I see stop. So that means my protein is done. And you could use either chart that you would like to use. You could use one and then check your work with another. But do make sure you're familiar with both charts just in case you ever did have to use one of them. Thank you so much for listening. If you have questions about transcription and translation, please send me an email.